The respiratory airway begins at the nose and mouth, continues through to the larynx, trachea, bronchi and bronchioles, and ends in the alveolar sacs. Human conducting airways are lined with an epithelium that consists of several morphologically distinct cell types with different functions. From the trachea to the major bronchi, ciliated, secretory or goblet and basal cells are present in the pseudostratified epithelium. The airway wall is comprised of surface epithelium supported by a reticular basement membrane, a submucosa, and a thin adventitial coat. The apical epithelial surfaces of the respiratory airways are coated by a mucus layer, a viscoelastic gel, which acts as a protective barrier and is mobilized by ciliary movement. Mucins, glycoproteins secreted by goblet cells, are a major component of the mucus. Goblet cells are found amongst the ciliary cells, mucus and extracellular matrix. These are calyx-shaped, polarized cells with a nucleus located basally and abundant secretory granules found in the apical region of the cell. The endoplasmic reticulum is widely distributed in the cytoplasm and includes thin tubular extensions forming a network among the secretory mucin granules. The Golgi apparatus, located in the supranuclear region, is arranged in a cup shape with an opening towards the apical portion of the cell. The Golgi apparatus is composed of cis, medial, and trans cisterns. Mucins are highly glycosylated proteins synthesized by goblet cells. These proteins are coded for by the MUC genes, which are transcribed by RNA polymerase II. The messenger RNA is processed in the nucleus before being transported into the cytosol through pores in the nuclear envelope. In the cytosol, ribosomes bind to the messenger RNA and proteins start to be translated. The signal recognition particle, the SRP, binds to an amino acid sequence that acts as a signal and stops translation. The messenger RNA ribosome SRP complex is recognized by a membrane receptor located in the rough endoplasmic reticulum membrane, to which the complex anchors. Once the SRP is removed from the complex, translation is resumed and the protein is simultaneously transported into the lumen of the rough ER. The signal peptide is removed. Once in the lumen of the rough ER, proteins accumulate in certain areas which, after evagination, are transformed into transport vesicles which carry the proteins into the Golgi apparatus. In the Golgi apparatus, proteins are glycosylated by the action of at least a dozen glycosyl transferases attached to the Golgi membranes. From the trans-Golgi, sections of membrane covered by clathrin evaginate. These vesicles contain mature mucins. Afterwards, secretion granules are formed by the fusion of many of these vesicles. After a maturation process, the granules are translocated to the apical plasma membrane through a network of microtubules. In this position, they are stored until exocytosis. Secreted mucins form a fundamental part of the mucus that lines the epithelium under normal conditions. In diseases such as bronchial asthma and chronic obstructive pulmonary disease, there is hypersecretion of mucus associated with an increased number of goblet cells. These are also increased by the action of cigarette smoke, irritant gases, and bacterial compounds.